Assalamualaikum and good morning to all viewers. Welcome to the National Institutes of Health CME session 2025, proudly brought to you by the Sector for Research Information Resources, NIH. The NIH CME platform is dedicated to showcasing the work of our researchers, offering a space to share their insights, experiences, and latest discoveries with the broader community. We are delighted to have you with us today. I'm Liyana from Institute for Medical Research, and I will be the moderator for today's session. But before we begin, let's take a moment to watch the IMR's corporate video.
The Pathological Institute of Kuala Lumpur was established in 1900 by the Resident General of British Malaya, Sir Frank Swettenham. Its aim was to promote the health status of the local population and to explore the great and unknown field of tropical medicine. The institute was renamed as the Institute for Medical Research, or IMR, a year later. Initially, the focus of its research was on beriberi and malaria. Subsequently, its scope expanded into the causes, treatment and prevention of a variety of other tropical diseases such as cholera, smallpox, leprosy, tuberculosis, typhoid, filariasis, dengue, melioidosis, scrub typhus, and nutrient deficiencies. With independence, the IMR became the research arm for the Ministry of Health Malaysia, where research conducted has been aligned with the country's health research priority needs. In more recent times, research at the IMR has progressed to gathering in-depth knowledge of allergy and autoimmune diseases, primary immunodeficiency diseases, and through the development of allergy test kits. Cancer research at the IMR has vigorously looked at diagnostics, prevention and early detection, as well as identification of novel biomarkers and targeted therapy for precision medicine. For this, the Institute has been equipped with a specific pathogen-free or SPF animal facility and biobank. The IMR has developed its expertise in nutritional biochemistry, where there is active involvement in preclinical studies on metabolic syndrome, diabetes, cardiovascular health, and endocrine-related diseases. The Institute's iodine test kit is used nationwide for monitoring under the Iodine Deficiency Program. The dawn of the next millennium saw many issues that were related to contaminants and pollutants in the environment and in the workplace and its impact on human health. Thus, in a forward-thinking manner, IMR conducted vulnerability assessment on impacts of climate change on health and health delivery. Malaysia is in a region that is full of biodiversity. The Institute has a unique interactive e-database called GlobinMed, which strives to become a referral source on traditional and complementary medicine, or TNCM. The Institute steadily has been moving towards drug discovery programs. The Center conducts preclinical safety evaluation of herbal extracts under Certified Good Laboratory Practice or GLP conditions, assessing efficacy of herbal products using in vivo as well as in vitro systems, phytochemical analysis, and structure elucidation. The IMR has continued to provide in-depth research related to infectious diseases, especially during outbreaks, and it is the reference center for Zika, dengue, influenza, leptospirosis, and melioidosis. IMR is the leading center conducting Wolbachia research in Malaysia. The Institute has equipped itself with a BSL-3 laboratory as a preparedness plan for any possible future outbreaks. Epidemiological research activities have helped to establish the distribution of diseases, its factors, and how it helps in controlling diseases. The IMR provides specialized diagnostic tests nationwide related to organ and blood stem cells transplants, diagnosis of inherited metabolic diseases, inborn errors of metabolism or IEM, genetic diseases, and multiple myeloma. In the international arena, the IMR serves as World Health Organization or WHO regional and collaborating center for infectious and tropical diseases. Under various programs, the IMR has produced a few patented products ranging from insecticides, fungi, dengue, chikungunya, saliva and forensic test kits, mosquito detector and trap, paint, anti-cancer toxin, and many more, from which some of the patented products have been commercialized. The 116 completed years of the Institute's illustrious service continues with new challenges and higher pursuits as the IMR transforms its biomedical facility to support research both nationally as well as internationally. IMR to be a world-renowned institution for biomedical research.
Welcome back. Just a quick reminder for those who just joined us, kindly register your attendance using the link in the description box. The participation certificate will be sent to you after the sessions. So if you have any questions or would like to share your feedback, feel free to drop them using the link provided and we'll go through them after the session. So let's get started with today's session. We've got an exciting topic to share today, looking at how metabolic surgery impacts the health of people living with obesity and diabetes. Given the global health challenges of obesity and diabetes, this study is both timely and highly relevant. So it is my pleasure to introduce our distinguished speaker, Dr. Fazliana Mansor, who is currently the research officer and the head of Endocrine and Metabolic Unit at the Nutrition, Metabolic and Cardiovascular Research Center in IMR. Dr. Fazliana holds a PhD in Biomedical Science from the Karolinska Institute, Sweden. Her research focuses on the molecular biology of endocrine and metabolic diseases, particularly on diabetes and obesity. She has also contributed to research exploring the therapeutic effects of traditional Malaysian herbs, such as Labicia pumila, or known as Kacip Fatima, in managing metabolic conditions. Today, she'll be sharing her insights on how metabolic surgery goes beyond weight loss to impact metabolic health at various levels. Her presentation is titled Metabolic-Bariatric Surgery in Obesity and Diabetes, Blood Profiles, Body Composition, Molecular and Metabolomic Impacts. Without further ado, let's welcome Dr. Fazliana to begin her presentation.
Thank you, Liyana. Assalamualaikum and salam sejahtera. I'm Fazliana from the Endocrine and Metabolic Unit. Our sharing session today is actually the summary of a project which we call Moment Study because of its very long title, which has been approved quite a few years ago in 21. However, we could only started to recruit in June 2022 due to COVID-19 pandemic. However, we managed to complete the study in 2.5 years. We closed the project last year. This is the presentation overview where I will explain about the background, types of surgeries, methodology, and we will discuss about the results and discussions based on the topics. Okay, before we go further, I want to explain on how we actually arrived to this study topic. In our unit, besides diabetes and endocrine-related research, we also focus on obesity research. So we do screening and intervention programs, which covers the school, workplace, and community. And maybe some of you already involved as participants in these projects. For laboratory analysis, we do biochemistry, which involve the blood profiles and also hormones. Also using molecular techniques involves genetics, epigenetics, and gene expressions. Now we just venture into metabolomics where we collaborate with others. And another aspect which is very important is we also use animal model in the diabetes and obesity study, especially to test on the natural products and functional food. So most of these are project collaborations with others. So we welcome collaborations with you if you're interested on these topics. Okay. Metabolic-bariatric surgery is gaining popularity as a weight loss solution. However, our understanding of the molecular mechanisms and metabolic pathways, particularly concerning local data, remains limited. But what is metabolic-bariatric surgery? It originated from Greek words where baros means weight and iatrika means medicine or surgery. It is often referred to as weight loss surgery, a surgical procedure designed to alter the digestive system to promote weight loss and improve obesity-related metabolic disorders. It is also considered when lifestyle changes like diet and exercise haven't been successful or when severe health problems due to weight are present. But why are these two terms used? Basically, bariatric surgery indicates variety of GI surgical procedures originally designed to induce weight reduction in morbidly obese patients. Because of the benefits, however, extend beyond weight loss and improvement in diabetes, hypertension, dyslipidemia, and reduction of overall mortality. So that is where the metabolic terms came in. So the use of metabolic and bariatric and metabolic terms are interchangeably used. And if you look at the latest literature review, metabolic bariatric surgery or MBS is used as a new term to describe both of these. Okay, this is the surgical intervention already included in the recent or the latest 6th edition CPG, Clinical Practice Guidelines, Management of Type 2 Diabetes Mellitus. You can check, it's available online on the criteria used for this method. And then this is even recent in 2023, the 2nd edition of CPG, Management of Obesity. The surgical procedures are also included in the CPG, where the Roux-en-Y gastric bypass, sleeve gastrectomy, and abdominal gastric banding are included. There is a list of criteria. Okay, just to introduce a few of types bariatric surgery, there is gastric sleeve, gastric bypass, gastric band, and intergastric balloons. Okay, these two, gastric sleeve and gastric bypass, are the most common bariatric surgery which are used worldwide and also in Malaysia. And we include these two in our study, even though we don't separate them into different analyses, but these are the two that we include them. First is gastric sleeve, where the new stomach is formed when the portion of stomach is removed. And the Roux-en-Y gastric bypass, or we just call it gastric bypass, is a weight loss surgery where the stomach is divided into a small pouch, and the small intestine is reconnected to this pouch by passing a portion of the stomach and upper small intestine. This limits the food intake and reduces nutrient absorption, leading to weight loss. The primary goals of our study are to assess changes in body composition, including the BMI and face angle before and after surgery. I will explain more on the face angle because that is a new topic. To evaluate biochemical markers such as HbA1c, hormones, adipokines, and inflammatory cytokines. Also to analyze molecular indicators like microRNAs and metabolomic profiles to elucidate mechanisms of diabetes remission. This is a prospective multi-center observational study for patients undergoing metabolic surgery. Our study involved private centers, which are, there's iHeal in Kuala Lumpur, Sunway Medical Center, and also KPJ Damansara. We recruited 111 obese patients who were already scheduled for metabolic surgeries. So we didn't intervene on the procedure. They were already in the list for surgeries. So we collect the data for this. Both men and women between 18 and 65. Okay, for the BMI of these studies, we choose the Asian BMI, which are above 25. If you see, this is quite different from WHO BMI. but this is for study purpose. And you will be surprised if using the Asian BMI, so many of us have obese and overweight criteria, maybe you, maybe me. So this is the time to take care of ourselves in terms of diet, nutrition and exercise and so on. But I will, today's session is more on the bariatric surgery. Then the patients were grouped into non-diabetes. They are obese but non-diabetes, obese pre-diabetes, and obese with type 2 diabetes. The diagnostic values for the pre-diabetes and diabetes is based on the HbA1c value according to the Malaysian CPG, where the normal is below, the HbA1c is below 5.7%, pre-diabetes is between 5.7 and 6.3, while diabetes is above 6.2%. For comparison, we also recruited healthy individuals between 18.5 and 22.9, about 26 individuals. Just the detail of inclusion and exclusion criteria. I'm not going to read the list. Okay, for the protocol paper, we already published one protocol paper for this study. Okay, this is just the overall flow of the study, where we have 3 time points, the important one: baseline, 6 months follow-up, and 12 months. Okay, but one interesting topic that I'm going to share with you later about the adipose tissue gene expression. We only collected the adipose tissue during the surgery. We don't do biopsy, during follow-ups. I think it's unethical at this point, but that is for the gene expression study. While the blood collection, we did 3 time points. The blood were processed into plasma and serum and analyzed for HbA1c, biochemistry, and all the blood profiles, ELISA for hormones, and the 2 microRNA analysis and metabolomics are the 2 topics. Even for me, it's new for us, so it's exciting to actually explore into that. Also, at these 3 time points, we measure the body composition using the body impedance analysis, or in short, BIA. Okay, another workflow. Start with recruitment, and then 3 time points, and then analysis. Even though it seems simple, 3 time points, but actually we did the recruitment for 1 year, and we managed to complete the follow-up. The list actually has been explained before. Okay, just another explanation about the healthy participants, and then we divide the obese patients into non-diabetes, pre-diabetes, and diabetes. Okay, why we choose adipose tissue for gene expression study? Okay, for this one, we didn't analyze pre and post, but we compare different groups of obese patients. Typically or historically, we know that white adipose tissue is important for storage, but now we have a lot more knowledge on it. It also controls metabolism through secretion of hormones, cytokines, proteins, specific lipid species, and microRNAs, which affects many things in the body. So the excessive accumulation of fat tissue in the obesity is the source of chronic low-level inflammation and can cause many issues. So I will share a diagram on the visceral fat. Okay, we do analysis on the anthropometry, body composition, HbA1c, blood profiles, and so on. Okay, the results today, I'm going to present according to this topic. We will start with anthropometric, then body composition, followed by blood profiles, molecular, and then metabolomics. Okay, we start with body composition, start with BMI. For this particular analysis, we only divide into 2 groups: non-diabetes, meaning they are obese but no diabetes, and diabetes. For both groups, significant reductions are recorded, and it's very significant. For the percentage body fat, the same trend also occurred where for both groups have a significant reduction with even less 10% from the baseline. Now, this is interesting. When we look at the skeletal muscle mass, okay, when we want to do the research, we were hoping that everything will go well. But for skeletal muscle mass, there are reductions, even within 6 months, and then some maintenance or some increase between 6 and 12 months, but not significant. Okay, I will explain more in that. Okay, what is visceral fat? There are different types in the body. Subcutaneous fat is available, or it is located under your skin. But visceral fat, omental fat, and also, those are the fats inside the abdominal, inside your abdomen. So these are the fat that we call it bad fat. So for both groups, both are significantly reduction, and that is good. Okay, now we look at the face angle. We know about BMI, we know about the skeletal muscle mass. This is a new, quite a new topic in medicine. It is measured with bioelectrical impedance analysis. The model that we use is quite different from usually used in the clinic. This one is InBody S10. It's a portable model, and it's used especially for renal patients, bedridden patients, but used for this study. It is particularly used for measuring the extra and intracellular water. It is considered as a biological marker of cellular health. The use of face angle as a predictor of diabetic complications and disease severity is of growing interest because due to its non-invasive nature, it also has potential to reflect changes in body composition, cellular integrity, and improve cell functions. So we get the figures from the body composition that we measured. Okay, this is the overall results of the patients, obese patients, without categorize them into different diabetes status. Significant reduction, 6 months, even further 12 months. That is good. But skeletal muscle reduce after 1 year, and then face angle also reduce, meaning that there is a loss of muscle and also reduce of this face angle value, because we expect that the better your body is, the higher the face angle figures or values. But the details of the study will be presented by Dr. Azlin in upcoming conference, so she will share more in that. So the conclusion about skeletal muscle mass and face angle: significant declines in both muscle and face angle occur within the first 6 months post-surgery, with minimal recovery observed at 1 year. The early post-operative phase is critical for muscle retention and cellular health. So our findings highlight the importance of targeted approach to prevent long-term muscle deterioration. So the doctors or the bariatric team have to emphasize on the exercise and nutritional aspects even before the bariatric surgery takes place. Now we go to the blood profiles, involve the cardiometabolic factors. Because our aim is to look at the HbA1c, we analyze according to 3 groups: non-diabetes, prediabetes, and diabetes. There are significant reductions in all groups, but when we look into detail on the magnitude of HbA1c changes over 12 months, the type 2 diabetes reduced more HbA1c compared to other groups. But this is logical because type 2 diabetes has high starting point compared to others, but this is good. However, when we look overall, of course, they are improved. And when you look at the 12 months, they cannot go lower because they already reached the normal values of HbA1c. Okay, now we look at the good cholesterol, HDL-C. Yeah, all groups showed significant improvement. But when we look at the magnitude, no significant difference in HDL-C. HDL-C, all groups, okay, this is already explained. The diabetes group had the greatest increase in HDL-C compared to prediabetes and non-diabetes, but it's not significant. But we can see trends there. And the details of these findings will be presented by Liyana in future conference, and we are also going to publish this. Okay, just the overall liver profiles, all groups improved. So the summary for the blood profiles, metabolic surgery significantly improves cardiometabolic risk factors in obese patients, particularly in those with type 2 within 1 year. All diabetes patients achieve HbA1c levels below 5.7%, returning to normal after 1 year. Most variables either improve or were maintained. However, the extent of changes was not statistically different between non-diabetes and type 2 groups. Okay, another aspect of blood component that we measure the hormone, namely augmentin. You might not be familiar with this because it is a newly identified secretory protein, also known as interleukin-1, selectively expressed in visceral adipose tissue relative to subcutaneous tissue. Okay, let's focus on let's focus on this chart. Obese group with prediabetes and obese diabetes have significantly higher levels compared to healthy and obese only. So the hormone might be an important indicator of diabetes in obese individuals. However, we did experiments, we did measure on the pre and post. We didn't find significant changes. There are trends, but not significant. So at this point, we are not sure whether there are really no changes or we don't have enough power for that. Okay, another hormone which is interesting and quite new is the ESM1 or endothelial cell-specific molecule 1, also known as endocrine, a protein encoded by the ESM1 gene released by vascular endothelial cells in several organs, may play a significant role in regulating cell adhesion and pathogenesis of inflammatory disorders. Despite its potential in these processes, because from the literature review that we found, like most of them are on cell culture and animals, so a lot more study have to be done on humans. The metabolic conditions, it has a relation, despite the roles in this process, the relationship between this hormone and metabolic conditions such as prediabetes and obesity is not well understood. So we did measure. I just want to summarize on this. Okay. We don't divide into different groups, just overall results. At 6 and 12 months post-metabolic surgery, there are a significant increment of ESM1, even when we compare prediabetes and non-diabetes. In obese patients, it is higher in the healthier group. Okay, let's look into inflammatory markers, because obesity and diabetes are associated with inflammation. We measured interleukin-6, MCP1, which is monocyte chemoattractant protein 1, and also TNF-alpha or tumor necrosis factor alpha. For all, they are reduced either at 6 or 12 months. There are some data actually unavailable here, only at 6 months, but we have good results for the reductions. Okay, now we move to molecular topic. The first one is gene expression, then I will explain on the micro RNA. The gene expression analysis was done on the adipose tissue, which was collected during the surgery. We compared the expressions between different diabetes groups among obese patients. Okay, if you just wonder, this one is the protocol of how we did the PCR, and this is the curve when we run the analysis. Okay, just to admit that the adipose tissue is one of the tissue that is quite difficult to handle. It's quite difficult to handle compared to tissue like muscle or liver. I think because of the fat content. So we extracted the RNA from adipose tissue and then convert to cDNA synthesis, run the real-time PCR, and analyze using the Gene Globe. That is a free software available online. Why I say it's difficult? First, just to get the high yield of RNA from adipose tissue, and the other one, we need a lot of RNA to start the cDNA synthesis. So you have to, even though you have the protocol from the manufacturer, you still have to optimize and do lots of experiments prior to that. Okay, this is human adipose tissue, which was collected during the surgery. So what we did, we brought the tube to the OT, to the operation theater, which was filled with liquid suitable for adipose preservation, for RNA and DNA and so on. So these are the team were extracting their RNA in the lab. Okay, don't worry about the small fonts, the small text, just examples of the results came out from that Gene Globe free software. So on the right is example of when the results has been analyzed. These are the results when we compare between obese only versus diabetes. Okay, still the same results, and there is actually a list of genes which are related to diabetes and insulin resistance. Okay, just to highlight, even this is quite small for me. Okay, the significant upregulated genes in prediabetes compared to non-diabetes. So most of the genes are related to inflammation and metabolism. Okay, I want to highlight this one. These are the example of genes both expressed in pre-diabetes and non-diabetes, meaning that these both group are obese, but one is when we check the HbA1c, the glucose, everything is normal. The patients are just obese. The other one is pre-diabetic. Pre-diabetic means it's not yet developed into diabetes, but the HbA1c level is already reached certain level. So some genes are already upregulated in obese individuals, although not pre-diabetic. What does this mean? When we are obese, although our blood profiles look okay, we go for medical checkup, we send our blood for laboratory analysis. No, the HbA1c, the glucose is okay, looks fine. So you thought you're just obese only. I'm fine. I'm still okay. But the gene expression in your adipose tissue already share same criteria like pre-diabetic gene expression. So at this point, healthy obese term should be used carefully, even though it is being used for cardiometabolic description. So this is important message to deliver. So the summary about adipose tissue gene expression analysis, some genes related to glucose metabolisms are already expressed in obese patients without prediabetes. I'm not talking about diabetes, but prediabetes. Although the glucose levels and HbA1c values in the blood do not reflect diabetes status at this stage, proactive actions should be taken at this point to avoid more serious conditions, because we want to avoid from diabetes progression. So metabolically healthy obese conditions should be defined carefully. But the limitations of this gene expression study, no samples were collected from healthy controls because they did not undergo surgery, so we assume about the disease progression. The samples only collected at baseline, so we cannot analyze before and after. So we conclude and we analyze as is. Okay, another aspect of molecular. This is, personally, for me, this is very interesting because I learn new things. This is the first time I venture or explore on microRNA levels. MicroRNAs are small, non-coding RNA molecules that regulate gene expression after transcriptions. They are small, single-stranded RNA molecules that are released into bloodstream and can be found in other body fluid. They are known to play a role in cell-to-cell communication and have been investigated as potential biomarkers for various diseases, including cancer, hope to be included into obesity and diabetes. When you, if we look at the literature review, one of the important terms or words are non-invasive. Of course, we still need to take blood for microRNA analysis, but it's still not as invasive because blood taking is quite normal in clinical setting. It's not as invasive as taking biopsy. So microRNA is quite like many research are into this. What we did, when we collect the blood, the serum is processed, then the microRNA is isolated using commercial kit. Then we quantify the microRNA, because only then we proceed to RNA PCR, then we analyze. Okay, still don't worry about all these small figures. But these are also the examples which came out from the gene globe analysis. I will summarize on the findings in next slide and another slide later. Okay, I'm not sharing with you with the 12 months post-surgery results, but for 6 months we have a list of overexpressed and underexpressed microRNA list. At post-surgery, we can already see the changes in the microRNA levels. Some gene expression could be altered since microRNA role is to regulate gene expression after transcription. Okay, I'm going to highlight these 2 microRNA which were in the figure before. For example, I think there's a typo there, but it's HSA microRNA 1955P, which is associated with hypertension with type 2 diabetes, and it is reduced. Another one is HSA microRNA 451A, which is related to associated with type 2 retinopathy. Okay, for this description, we don't do the comorbidity or complication study. We get the data from the library freely available in the database in the internet. So microRNAs have potential to be type 2 complication early diabetes. And from our study, we clearly see that, before and after there already changes. So even though you can see the physical body composition, blood profiles like HbA1c, but at cellular level, they are already changed. Okay, this is another new area that I that we venture. Specifically for this, I would like to thank our collaborator, Dr. Naim, for collaborating with us, because he is the key person for this analysis. So metabolomics is a rapidly evolving field of life science that uses advanced analytical chemistry techniques in conjunction with sophisticated statistical methods to comprehensively characterize the metabolome. It can differentiate different stages of diabetes, and I will not read the rest, but my hope is that even though now it's not accessible to public and the equipment is not available in many places, in future it is very potential for precision medicine, because it can detect, it can see changes of individuals' metabolites. We use the untargeted approach using the 600 megahertz nuclear magnetic resonance NMR spectroscopy. The equipment is available in our NIH IMR shared facility, BioRCF. We analyze all samples, healthy controls, obese, pre-diabetic, non-diabetic, and diabetic. Sorry, like too many words here. Obese, pre-diabetic, non-diabetic, and diabetes. So basically, we analyze all groups. Okay, the figures at the bottom here, yeah, just the process of the flow of the sample processing even before analyzed in the NMR. So the sample processing and the sample collection, which tube to use. Yeah, those are the things that is very important to ensure that the final results are clean and easily analyzed. Okay, just another workflow. So we measure the obese patients and healthy controls, prepare the samples, and then inject it into the NMR. So these are the software that we use, namely Kinomics or Chinomics, MetaboAnalyst, and SIMCA. Okay, the results of OPLS-DA all groups to healthy controls. Let's see one by one. We start with obese non-diabetes. This is obese only. So we compared 3 different status of diabetes in obese patients compared to healthy controls. Okay, this is obese only, so there are still some metabolites overlapping. For pre-diabetes, you can see that they are start to move away. And for diabetes, okay, so obese, diabetes, and healthy patients, healthy means they are healthy, no comorbidity, not obese, so their metabolites are very, very separated from healthy obese. From healthy patients. Yeah. If you're familiar with PCA, that is principal component analysis. OPLSDA is quite the same thing. Okay, just a few more slides to finish. Okay, another component that we analyze is the correlation heat map. We found 4 clusters, and each clusters are exhibited strong correlations among their respective metabolites. Okay, this is, we're going to publish this soon because we just received the DG's approval last week. So hope everything will go smoothly. And even this one is quite fresh data. If previously we only compared different groups of obese diabetes status, now is we compare overall between pre and post. From volcano plot and t-test, we found a list of metabolites. Oh yeah, the OPLSDA is the orthogonal projection latent square discriminant analysis, quite long. This is quite similar to PCA, but a bit more complicated. We also formed the heat map correlation. So the conclusion from that is metabolic profiles in obese patients are significantly different from healthy body weight, BMI, the adipose tissue gene expression levels, especially in prediabetes, need attention to prevent diabetes from developing. So now we know, even though it's quite difficult to actually capture prediabetes because there are so many people walking around pre-diabetic. So these are the group that need to be tackled. Bariatric surgery significantly have effect on weight reduction, resolution, and improvement of obesity-related conditions like diabetes and metabolic syndrome. Targeted approach are essential to prevent long-term muscle problem. So exercise and nutritional aspects need to be focused. The changes at molecular levels and metabolomic profiles can be observed after the surgery. So basically improvement on body composition and cardiometabolic factors. So what's so significant about MOMENT study? This is particularly significant for Malaysia, especially for local data, because metabolic surgery is not yet widely adopted. We provide data for that by providing comprehensive data on anthropometric, biochemical, and molecular changes post-surgery. The study aims to inform clinical practices and potentially support the broader implementation of metabolic surgery as a treatment option for obesity type 2. Another conclusion, the message that you can bring home, can take home. So our future plan is to carry out another study in government facilities, because the one that we did was in private practices, but this time is to evaluate effectiveness of weight loss program, where they have weight loss program before the bariatric surgery, which involves exercise and also diet. And also we want to explore and expand more on gut microbiome, targeted metabolomics, because the one that we had results was untargeted, and if possible to explore more than epigenetic studies. So this I would like to thank our surgeons as our collaborators and our investigators from various private practice, a line of research and medical officers who work dedicatedly for the project, and of course our medical lab technologists who work tirelessly and very committed to the project too, and our personal MyStep personnel since the project began. Okay, with this I thank you, and these are some photos I want to share of our journey. Okay, with that I thank you.
All right, thank you so much, Doctor, for the insightful presentations. So it's clear that metabolic bariatric surgery influences far more than just weight. We have seen its significant effects on blood biomarkers, body composition, and even on the molecular mechanisms. So I'm sure everyone gained new understanding on its role in treating diabetes and obesity. Now we have come to Q&A sessions. We have received several questions from our audience. So let's start with the first one. So the first question is, keeping participants engaged over time is always tough in longitudinal studies. Did you experience any dropouts?
Yeah, actually, yes. We experienced dropout either at 6, 12 months, and even both. So that is important to take into consideration when we do the analysis.
Okay, thank you, Doctor. So that highlights the importance of also follow-up planning in the clinical research, right?
Yeah.
So, all right, moving on to the next questions. Could you elaborate on the statistical methods that you use to ensure robustness of your findings?
Is there any more question? So that is on the statistical.
Yes.
Okay.
So that's on the statistical part.
We have different aspects of the studies, different aspects of the studies. So each has its own way to be analyzed. For example, the anthropometric and biochemistry or blood profiles, we use GEE. GEE is generalized estimating equations, which take into account all the participants, whether they have missing 6 or 12 months follow-up. So that is one. Another one is, for example, for molecular studies, we use, it's not manually calculated. So basically we follow the protocol, and then we prepare the data and we submit into the software. So that is where they analyze in terms of what genes are or microRNA are upregulated or downregulated. But it's important to have clean data, good data when you submit. And the other one is Metabolomics. That is also a different one. When I mentioned about heat map analysis, so that is on either use genomics, and also because there are 3 component genomics, metaboanalysis, and the other one is can't remember now, but I will mention again later.
So it's in the slide, right?
Yeah.
So now we have questions on the molecular part. So measuring microRNA can be tricky compared to DNA and RNA. Can you explain how you measure it in your study?
Okay, since I'm moving the slides, let's go to this. Okay, the slide looks very simple, but actually it took us about half a year to optimize. Yeah. So when the serum is collected, then we extract the microRNA. It has to be measured. It has to be to make sure that the quality is good. So we use the qubit microRNA assay. That is one, because you cannot measure just using simple nano drop. Yeah, that is a type of spectrometer. So we use qubit.
All right,
Yeah,
I
and
think
it has to be optimized.
all right, that gives a good sense of challenges and solutions when working with microRNA.
Yeah.
I think.
So maybe I want to explain more about this. When you, I think if anyone want to venture or go into this area, first just to optimize and take your time. Do not don't rush, because I think as we know in molecular, things are quite expensive nowadays. So it's important to focus on what you want and then optimize one step at one point before you move to other steps.
All right. I think we can read another, the last questions.
Okay.
So the next question is, given that the study was conducted in private healthcare settings, do you think the result would be dissimilar if it were conducted in government hospitals?
Okay. Before that, I want to maybe share a bit about the history of the study. It's supposed to be done in an HKL, but then the pandemic COVID-19 hit. So we moved to private practice. Because during that time, the surgery changed from elective to non-essential. So we go to private practice. So maybe there is a difference. But at this point, I cannot say because we don't have the data. This is the data that we have now, according to their study flow. According to their clinical flow. We might have, I don't know, maybe better data or maybe different data if we do it in the government practice. Just like, as I mentioned in the future plan. So that will be very interesting.
All right, we're looking forward for that study. Okay, Doctor. Okay. Due to time constraints, we have reached to the end of today's sessions. I would like to thank our speaker for sharing such a valuable insights from the Moment study. Hopefully, this session has shed light on how metabolic surgery can improve the health outcomes of obese patients. As we wrap up, let's remember how crucial collaboration is in translating research into practice. Together, we can continue to advance patient care and metabolic health. On behalf of organizing team, thank you for being with us, and we look forward to seeing you again in the future session. Take care and stay productive.